So today I will discuss on digestion and absorption of lipid. We all know that the major dietary lipids are triacylglycerol, cholesterol, and phospholipids. The average normal Indian diet contains nearly 20 to 30 grams of lipids per day, while the Western diet has a bit increased concentration of lipids. So we will see digestion of lipids starts at a very minor level right from the mouth, followed by stomach, and the majorly it happens the in the small intestine. So we will see the various events in the mouth following once we chew the food. So due to the mechanical digestion, there happens the mixing with the saliva, and there is a limited, very limited enzymatic digestion takes place under the influence of lingual lipase, and these enzymes are being secreted by the von Ebner glands of the lingual region. And because the stay of food in the mouth is for very less time, so enzyme work at the optimum pH of 2.5 to 5. So along with the food, the enzyme is also mixed and it shifts itself right from the oral cavity to the stomach. So in the stomach, the following events takes place like mixing and churning. Then here there is a secretion of gastric lipase under the influence of gastric hormone, and then this mixing and churning will facilitates the breakdown of bigger fat molecules or droplets into the smaller one, and the lingual lipase from the mouth enters the stomach along with the food. As we have already told, the pH is 2.2 to 2.5 to 5. Here also this lingual lipase is active. The It acts on the short chain triglycerides. Short chain triglycerides are present in the milk, butter, and ghee, and the action of this lingual lipase is observed to be more significant in the newborn infants. So, and the gastric lipase is acid stable with an optimum pH of 5.4, and it is secreted by the chief cells, and the secretion is stimulated by the gastrin. So nearly 30% of digestion of TGs occurs in the stomachs. Then further from the stomach, there is a shifting of food into the duodenum, the first part of the small intestine. So in the intestine, the emulsification is the prerequisite for the digestion of lipid, and the lipids are dispersed into the smaller droplets, so due to which the surface tension is reduced and the surface area of the droplets is increased, and this process is favored by the bile salts peristalsis, and the phospholipid. Then the bile salts are important for digestion of lipid. The bile salts are present in the bile and which lower the surface tension, and they emulsify the fat droplets, and emulsification increases the surface area of the particles for enhanced activity of the enzyme. So once the food enters here under the influence of enzyme enterokinase, there occurs the secretion of cholecystokinin, which stimulates the pouring of the bile contents by the gallbladder into the small intestine. Similarly, the secretin hormone, it stimulates the secretion of the pancreatic juice into the small intestine. So this pancreatic juice contain the pancreatic lipase, while the bile juice contain the bile salts. And due to the action of this bile salt, there is a emulsification which facilitates the further digestion and absorption of fats. So then the lipolytic enzyme in the intestine, so they are pancreatic lipase, Here there is a pancreatic lipase. Along with the pancreatic lipases, there is a colipase, which facilitates the action of pancreatic lipase. Then there is a cholesterol esterase, and then third enzyme is phospholipase A2. So these are the major enzymes present into the pancreatic juice, and they all facilitate the complete absorption, complete digestion of fats. Then the bile entering into the duodenum serves to neutralize the acid chyme from the stomach and provides a pH favorable for action of pancreatic enzyme. That is around 7.7. So here there is a change in pH right from acidic to basic. The pH is now 7.7, which favors the action of pancreatic enzyme. Then here, the first of all, there is a digestion of there is a digestion of triglyceride. So with the help of pancreatic lipases, there occurs the deesterification right from first to third carbon of the glycerol, forming the two monoacyl glycerols. Here occurs the formation of two monoglycerols, and then an isomerized shift the bond from the position 2 to 1. That we will tell you later on in detail. The bond, then the cholesterol simultaneously, the cholesterol ester may hydrolyze to free cholesterol and fatty acids. The action of phospholipase A2 produces lysophospholipids. So what are the things present into the digesta? They're the major end product in the digesta are two monoacyl glyceride, that is around 78%. Then we have one monoacyl glyceride, that is 6%. Then glycerol is available than fatty acids. And thus digestion of TAG is partial, that is incomplete, because not it is still in the form of one or two monoacyl glycerols. Then the colipase, which is being secreted along with the pancreatic lipase. So the binding of colipase to the triacylglycerol molecule at the oil-water interface is obligatory for the action of lipase. The colipase is secreted by the pancreas as an inactive zymogen, and it is activated by trypsin. Fine. So here there is a more clear account of that large fat droplets are broken up into the small fat droplets after being emulsified with the help of bile salts. Then further, what happens? These are the large fat droplets are emulsified by the bile salts, and this is the lumen of intestine. And then this further, with the action of a lipase enzyme, which I have already told you in the previous slide, this is triacylglycerol. This is the glycerol backbone, and these are the ester with the fatty acids. So lipase breaks down these long chain fatty esters bonds, and leading to the formation of just one ester bond is there. Which is attached with the glycerol. So this is known as monoacyl glycerol. It may be two monoacyl glycerol, or if by the action of isomerase, this ester group is shifted to position 1. So it becomes the one monoacyl glycerol. So this is how the enzymatic digestion by the pancreatic lipase happens. Then after being digestion, the absorption of products of fat digestion takes place. So here what we will see is the fatty acids and monoglycerides resulting from the fat digestion leave the micelles and enter the epithelium cells. So after being emulsified and being digested, there occurs the formation of micelles. Micelles, what are the micelles? And these are a kind of association molecule which are interiorly they are more hydrophobic, and outwardly they are hydrophobic. So they facilitates the absorption of fats right from the intestinal lumen into the intestinal epithelium. So the only function of this, these myceles, these temporary myceles facilitate the absorption of these fat molecules. So the product of the digestion, namely the 2-monoacylglycerol, long chain fatty acid, cholesterol, phospholipids, and the lysophospholipids are all incorporated into these molecular aggregates and to form the mixed myceles. So that's how the mixed myceles are being formed. And the myceles are spherical particles with an hydrophilic exterior and the hydrophobic interior core. And due to their detergent action, the bile salts help to form the micellar aggregates. And this micellar formation is essential for absorption of fat-soluble vitamin as well, such as vitamin A, D, E, and K. So the myceles are aligned at the microvillus surface of the jejunal mucosa. So this is all jejunal mucosa, and the fatty acid, the 2-MAG, and other digested products, they all are passively diffused into the mucosal cell. This is the mucosal epithelium. So, and leaving behind the what it is left here, leaving behind the bile salts. So, what is happened of the bile salt? Because bile salt is not going to be absorbed inside the mucosal cell. Rest of the contents get inside the cell, while the bile salts are remain in the lumen. So, these bile salts are they go for enterohepatic circulation. What is the enterohepatic circulation? Actually, the bile salts are left behind, which are mostly absorbed from the ileum and return to the liver to be re-excreted. And about 90% of dietary lipids are normally absorbed. So this is the enterohepatic circulation. Further, inside the mucosal cell, there is a re-esterification and reformation of the compound, these triglycerides and the cholesterol esters and phospholipids. So the fatty acids are first activated to fatty acyl coenzyme A here, and by the action of enzyme synthetase or thiokinase, and this process needs the lysis of two high energy bonds from the ATP, and that two such activated fatty acids react with the monoacylglycerol to form the triglyceride. Then majority of molecule follow this MAG pathway. Then free glycerol is not involved in the formation of triglycerides, but the cell can convert glucose to glycerophosphate and then add 3 molecule of acyl group to synthesize the triacylglycerol. So this is how there is a generation of triacylglycerol. Then short chain fatty acids are not re-esterified inside the mucosal cell. Instead, these are directly absorbed into the bloodstream. So there is no obstruction in their passage. Right from the mucosal fluid to the blood, they can easily be absorbed through simple diffusion and reach into the directly blood circulation. So now, so what is happening here? But there is a problem with the assimilation, absorption, and assimilation of these reassociated fats. These are the huge molecules. Moreover, they are hydrophobic, cannot be soluble or miscible with the blood. So they need some vehicle for their further transportation into the blood. So that's how there is a generation of chylomicron. So fatty globules combine with the protein to form the chylomicron. So these are the amphipathic vehicles. With the help of Golgi apparatus and endoplasmic reticulum, there occurs the formation of these associated molecule, ball-like structure. These chylomicrons are extruded from the epithelial cells and enter the lacteals. A lacteal, that is a lymph capillary, and then lymph inside the lymph, the lacteal transport the chylomicron away from the intestine. Further, all these lacteal contents are poured into the thoracic duct, and then from the thoracic duct it's all dumped into the subclavian vehicle, vesicle. So this is how there is a indirect pouring of these chylomicron right from the lymph into the blood circulation. So this is the structure of chylomicron, one of the important lipoprotein which are generated in the intestinal mucosal cell. And as I have already told you, they consist of outerly hydrophilic portion and its interior is hydrophobic. So these are the embedded apoprotein. So apoprotein B48 is there due to the incomplete no expression of 48% expression of consent protein. So this is known as apoprotein B48. Then phospholipids are outwardly arranged. The hydrophilic heads are outwardly directed, while the long chain fatty acids are interiorly directed. And this is the inside, all TGs are embedded inside this. So this association whole molecule is known as chylomicron. So what is the key message from the lecture? In the stomach, the fat is separated from the other food substances. And in the small intestine, the bile emulsifies the fats while the enzymes digest them. The intestinal cell absorb the fat. The long chain fatty acids form a large lipoprotein structure called the chylomicron that transports fat through the lymph system. Then chylomicrons are formed in the intestinal cell and carry the lipids from the digestive tract into the circulation, while the short and medium chain fatty acids can be absorbed directly via simple diffusion into the bloodstream from the intestinal microvilli. And because they are water soluble, so then cholesterol absorption is hindered by food high in the fibers. When the energy supplies are low, the body utilizes stored fat reserves for energy purposes, which is the second important source of the TGs are the second important source of energy after the glucose. So that's all about the digestion absorption of fats. The little bit clinical aspects. There is a due to there can be two conditions. Either can be the defective digestion or can be the defective absorption. So if the digestion is defective, it may be like in steatorrhea. Steatorrhea is a clinical condition. There is a daily excretion of fat in the feces more than 6 gram per day, and it is due to the chronic diseases of pancreas. So in severe pancreatitis, there is unsplit fat is seen in the feces. This is the characteristic of the feces in the person suffering from steatorrhea, because the pancreatic lipases are not there, not available for digestion of the fats. Second condition can be defective absorption, means digestion has happened properly but there is a defect in the absorption. So in such condition, if the absorption alone is defective, most of the fat in the feces may be split fat and fatty acids or monoacylglycerol. And defective absorption may be due to the celiac disease, sprue, Crohn's disease, or it can be due to the surgical removal of intestine, or it can be due to the obstruction of bile ducts, which may be due to the gallstone, tumors of head of pancreas, or enlarged lymph glands. So this is how we can, just by testing the available fats present into the feces, we can differentiate whether it is the defective digestion or the defective absorption of fats. Thank you so much.